Sds Page stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. This is a laboratory technique used to separate proteins based on their molecular weight. Let's begin with the structure of proteins. Proteins are complex folded structures made up of long chains of amino acids called polypeptides. These folded structures are stabilized by various interactions, including hydrogen bonds, disulfide bridges, and hydrophobic interactions. In Sds Page, chemicals like Sds and beta-mercaptoethanol denature proteins, disrupting those interactions and coating them with a uniform negative charge. This groundbreaking method of protein separation was first described by Ulrike Lemeli in 1970. So how do we carry out the procedure? First, a loading buffer is prepared. It contains SDS, bromophenol blue, beta-mercaptoethanol, and glycerol. This loading buffer is then added to the protein sample. Protein samples may be biologically derived, for example, from eukaryotic or prokaryotic cells, tissues, environmental samples, or even purified proteins. After mixing the protein with the buffer, we heat it at 95 degrees Celsius for 5 minutes. SDS is an anionic surfactant. It has a polar head group with a net negative charge attached to a long hydrophobic carbon chain. SDS denatures native proteins by disrupting hydrogen bonds, hydrophobic interactions, and ionic interactions. The reducing agent beta-mercaptoethanol is used to break disulfide bonds. When the protein mixture is heated, the hydrophobic tail of SDS binds evenly along the protein chain while the negatively charged head stays on the outside, giving the entire protein a uniform negative charge with a linear structure. Next is gel preparation. Solutions for separating and stacking layers are taken. Both solution contains acrylamide, bisacrylamide, ammonium persulfate, TEMED, but at different proportions. First, the solution for the separating layer is poured between the polymerizable glass plates where it settles at the bottom. After that, the solution for the stacking layer is poured. It forms a layer on top of the separating gel. Acrylamide is the main building block that polymerizes to form long chains. Bisacrylamide is a cross-linker that connects acrylamide chains, forming a 3D mesh. Ammonium persulfate generates free radicals which start the polymerization. TEMED speeds up the production of free radicals from ammonium persulfate. This helps the gel solidify faster. In the top layer of the stacking gel section, lower concentration of acrylamide results in large pore size, and for the bottom layer, higher acrylamide concentration results in small pore size. A plastic comb is placed on top of the stacking gel during polymerization. This creates small wells in the gel where the protein samples will be placed. Once the gel is fully polymerized, the comb is carefully removed. The gel cassette is placed vertically between two electrodes. The positive electrode is located at the bottom of the gel, while the negative electrode is positioned at the top. The setup is then inserted into the electrophoresis chamber. Triglyceride chloride is poured into the electrophoresis chamber to enhance the electrical conductivity through the gel. The protein sample mixed with the loading buffer is then placed into the wells of the stacking layer of the gel. After that, a voltage source is connected to both electrodes to begin the electrophoresis process. Due to the applied electric field, negatively charged polypeptide chains start going towards the positively charged anode. Due to the larger pores in the stacking gel section, this layer does not separate the proteins. Its job is to stack them. That is, gather all the proteins into tight, narrow bands so that they can enter the separating gel together. The bottom layer has smaller pores. So when protein goes through this layer, smaller pores stops them at different positions based on their size. Bromophenol blue used in the loading buffer will work as a tracking dye which will say when to stop the experiment. Due to its relatively small molecular size, bromophenol blue migrates faster than the proteins. By visually tracking this colored band, we can monitor the progress of electrophoresis. This allows us to stop the voltage before the dye and the protein samples have completely migrated through the gel. Once the voltage run is complete, the gel is removed from the glass plates. The stacking gel, which no longer holds useful information, is discarded. The separating gel is then stained using Kumazi blue dye to visualize the protein bands. After a period of staining, the gel is washed to remove unbound dye, revealing distinct blue bands. each one representing a specific protein size in the sample. By comparing these bands with a molecular weight marker, we can estimate the approximate size of the unknown proteins.
One of the major real-world applications of SDS Page is in HIV testing. HIV testing is done using the Western blot technique. The first step of this technique is separating the protein. This step is done through SDS Page. Here, HIV proteins are first treated with SDS. SDS lyses or breaks down the proteins in HIV virus like P24, GP41, and GP120 and coats them with a uniform negative charge. After placing the solution in the electrophoresis chamber, the proteins get separated based on their size. The second step is visualizing the protein bands. So, after separation, these proteins are transferred to a membrane. Human serum contains antibodies. After centrifuging the blood, the separated serum is added to the membrane, and if HIV antibodies are present, they bind to specific proteins. A secondary antibody is added to make these bindings visible as color bands, confirming HIV infection.